morning everyone. Welcome to Media Center, Dr. V.S. Krishna Government Degree College, Autonomous, Visakhapatnam. I am GRNS Sujatha, lecturer in Botany, Dr. V.S. Krishna Government Degree College, Autonomous, Visakhapatnam. Today we are going to discuss about the chromosomes, its structure, introduction, structure of chromosomes, types of chromosomes, chemical composition of chromosomes and eukaryotic chromosome structure. Chromosomes are the physical basis of the heredity, and these are the rod shaped stained bodies which are present in the nucleus of the cell. So, these are made up of by the DNA and proteins, and they play a vital role in the hereditary variations and mutations and development of evolutionary development of the species. So, now let we see the history of the chromosomes. The chromosomes were first observed by the von Nageli in 1848. So, then the Fleming, he observed the chromosomes and used the term chromatin, that is the colored part which is present inside the nucleus, he named it as the chromatin. Then Waldeyer, he explains that the color shaped bodies as the chromosomes. Color shaped bodies as the chromosomes. Chroma means color and soma means body. These color shaped bodies are named by chromosomes by Waldeyer. Then Sutton and Boveri, in their chromosomal inheritance theory, they suggested that chromosomes are the physical basis of the heredity. Then Kaufman and others, they explains the morphological characteristic features of the chromosomes. Then Korenberg in 1974, Korenberg, he explains the internal arrangement of the DNA inside the chromosomes by the nucleosome model. This is regarding about the history of the chromosomes. Now we are going to see about the size and number of the chromosomes. The size, the chromosomes are varies from 0.5 mu to 3 mu in their length and 0.2 to 0.3 mu in diameter. Generally, the plants have the large chromosomes than the animals, and dicots have more number of chromosomes or high number of chromosomes than the monocots. And the chromosome number is specific for a given species. That means each species have a specific number of chromosomes. Suppose you see Homo sapiens, human beings have the 23 pairs of chromosomes, onions have the 24 pairs of chromosomes. That means each given species have a specific number of chromosomes. The number of chromosomes in a somatic cell is noted as the chromosome number, that is denoted by the 2n, and the haploid set of chromosome is also known as the genome. The chromosomes, autosomes and allosomes. So, each diploid cell consists of two sets of chromosomes. Among these, two types of chromosomes are there, autosomes and allosomes. Autosomes are named as the somatic chromosomes, means they are not involved in the sex determination. Allosomes are mostly one pair of chromosomes, they are involved in the sex determination of an organism. So, these are called as the allosomes. In human beings, XXX represent the female and XY represent the male. Now, we are see the structure of chromosomes. So, generally the chromosomes appears the chromatin thread like structure during the interphase and resting phase and the permanent cells. But during the meiotic divisions and the late prophase stages only the suitable stages for observing the chromosome structures. In the meiotic phase, in the mitotic metaphase, chromosome shows the following structures. Those are the chromatids, centromere, telomeres, secondary constriction and chromomere structures. Commonly these 5 following structures are observed in the mitotic metaphase chromosome. So, this is the structure of chromosome. This chromosome consists of, this is the chromatid. The chromosome consists of two arms, P arm and the Q arm. So, the chromatid is produced by the replication of the prophase chromosome, and it forms the two symmetrical parts. These are called the chromatids. So, these chromatids are together at a single point, that is called the centromere, and each chromatid have the single DNA molecule. So, the long string like DNA molecules are formed and compact in the chromatid structures. So, in metaphase chromosome consists of two symmetrical structures. These are named as the chromatids. So, chromatids are the homologous chromosomes. Homologous chromosomes means that among the two sets of chromosomes, each chromosome in pairing, these two are similar in their structure, gene content and gene composition and gene arrangement. These two are named as the homologous chromosomes. The chromatids formed from the homologous chromosome is called the sister chromatids. Is called the sister chromatids. So, these two chromatids are called the sister chromatids. These two are the homologous chromosomes. These two are similar in their physical structure and gene composition and gene content. So, the chromatids are formed from a single chromosome is called the sister chromatids, and these two are called as that means the chromatids are formed from the homologous chromosomes is called the non-sister chromatids, sister chromatids and non-sister chromatids. And these two chromatids are joined at a center point, that is called the centromere. So, in metaphase chromosome, it consists of two symmetrical structures called the chromatids. Each one consists of the single DNA. The chromatids which are formed from the single chromosome, these are called the sister chromatids. The chromatids formed from the homologous chromosomes, it is called the non-sister chromatids. Homologous chromosomes means in each somatic cell, the chromosomes are in two sets and each one in pair. So, these two chromosomes are similar in their structure, gene content, gene composition and gene arrangement. So, these are called the homologous chromosomes. The chromatids which are formed from the homologous chromosome is called the non-sister chromatids. So, centromere is the region which two chromatids are attached here. The centromere is the where the two chromatids are attached during the mitotic division. This centromere also called as the primary constriction. During cell division, the spindle fibers are attached to this centromere, and in the anaphase, the chromosomal movement takes place through this centromere only. So, because of its function, it is named as a kinetochore. So, types of chromosomes depend upon the location of the centromere. The chromosomes are classified into different types, that is the metacentric chromosome, submetacentric chromosome, acrocentric chromosome and telocentric chromosome. Metacentric chromosome means the centromere located at the center of the chromosome, that is called the metacentric chromosome. If the centromere is located near the center, that is called the submetacentric chromosomes, and the acrocentric chromosome, in these chromosomes the centromere located near to the tip of the chromosome, that is called the acrocentric chromosome. That is the telocentric chromosome. That means the centromere attached at the telomere region, that means only single arm is seen in that chromosome. These are the different types of chromosomes by the location of the centromere, that is metacentric, submetacentric, acrocentric and telocentric. And also the number based on the number of the centromere also we are classify the chromosomes into different types, that is the eccentric chromosomes, holocentric chromosomes and dicentric chromosomes. Eccentric chromosome means the chromosomes without centromere is called the eccentric chromosome. Suppose diffused centromeres are, that means the number of centromeres are located on the chromosomal region, that is called the holocentric chromosome and dicentric chromosome. Sometimes the chromosomes have the two centromeres, these are named as the dicentric chromosome. So, the centromere is made up of are constructed by the DNA, repetitive DNA and some proteins. They are arranged in 3 layers and form the disc shaped structure. So, the main function of this centromere is it gives a space to assemble the microfibrils during the cell division, and also it is help to form the metaphase plate during the mitotic division at the metaphase. The next another important part of the chromosome structure is the telomeres. The two ends of the chromosomes are called the telomeres. The two ends of the chromosomes are called the telomeres. So, these telomeres shows the polarity. These telomeres shows the polarity, and also they prevent the joining of other chromosomes. These prevent the joining of other chromosomes and they protect the integrity and the individuality of the chromosomes, and it also prevent the joining of other chromosomes to this, and it prevent the telomeric region, and it prevent and protect the chromosomes individuality. At the molecular level, how the telomeres are look like means, the molecular level, the telomeric region of the chromosome is operating by the sequences of 3' to TTGG to 5'. This is the molecular sequence of the telomeric chromosome region, that is 3' to TTGG 5' direction, they are seen in the telomeres. Now, you see another important part of the chromosome is the secondary constriction. In addition to the primary constriction, that is called kinetochore or centromere, in addition to this, another type of secondary constrictions also seen in the chromosomes. These are called the secondary satellite chromosomes. So, this is the secondary constriction and this is the primary constriction. So, how it differs from the primary constriction means, how it is differ from the primary constriction means, they are not binding to the chromosomal segment during the anaphase, and it is the region between the telomere and the secondary constriction is called the satellite. These chromosomes are called the satellite chromosomes. These satellite chromosomes are closely associated with the nucleolus, and in each somatic cell nearly 1 or 2 satellite chromosomes are present in each somatic cell. So, this is the satellite chromosomes and secondary constriction. Now, the chromomeres. The chromomeres are the structures which are seen in the metaphase chromosome. In the mitotic division, a small bead-like structures appears on the chromosome. These are named as the chromomeres. These chromomeres are nothing but the nucleoproteins. It is believed that genes are located within the chromosomes, that one chromosome may possibly represent the single gene. So, see here the chromomeres are completely made up of by the nucleoprotein substances. Now, the karyotype. The term karyotype has been given to a group of characteristics that are identified by a single set of chromosomes, that is named as the karyotype. The diagrammatic representation of karyotype is called the ideogram. Now chromatin material. So chromatin material, that is the present inside the nucleus during the interphase, it is look like in different colors. Some part is highly colored part is seen and some is slightly colored region also observed. So you see the chromatin is classified into two main categories, that is the heterochromatin and the euchromatin. Heterochromatin is a dark stained condensed region of chromatin is called the heterochromatin. It is completely made up of by the repetitive DNA, and these are the condensed portions are named as the karyosomes. Euchromatin is the portion of the chromatin material which is slightly stained, only they are partially condensed. Suppose you see the heterochromatin is the highly condensed part and the euchromatin is the slightly condensed part. So the euchromatin contains the structural genes which replicable and transcribed during the G1 phase of the interphase. That means the structural genes are present in the euchromatin and the repetitive genes are present in the heterochromatin. The chemical composition of the chromosomes. The chemical composition of chromosome consists of the DNA, proteins and RNA. In the chromosome, 30 to 40% of DNA and 40 to 60% of proteins and 1 to 10% of RNA is seen in the chromosome. So among these, DNA is the most important chemical component of the chromatin. It plays central role in the controlling the hereditary. So it is a genetic material and it controls the hereditary. So the DNA present inside the chromosome is two types. One is the unique DNA and second one is the repetitive DNA. Unique DNA is the repetitive DNA. Suppose you see the chromosome, in this picture you observe the chromosome and the DNA, how the DNA is arranged in the chromosomes. So unique DNA means the DNA segment whose base sequence are present only in a single copy, they are not replaced in the genomic area. That means only single copies, that is all the structural genes, histones, RNA are made up of by this unique DNA. That means the unique DNA is specific, it is only present in a single copy. Next, repetitive DNA. Repetitive DNA sequences are the few to several hundreds of base pair and they are in the several multiple copies. So unique DNA is a single sequence and the repetitive DNA is the multiple copies for the genome. So in this picture you observe this is the how the DNA is completely arranged inside the chromatin material, how the genes are located and how they are arranged in the chromosome. Next, proteins. The proteins present in the chromosome, these are divide into two. Histone proteins are the non-histone proteins. Histone proteins are the basic proteins. These basic proteins are enriched with arginine, lysine being the basic histones. So that means they have the basic amino acids and they are tightly bind to the DNA. So histone proteins have the basic amino acids and these are the basic proteins and they are tightly bound to the DNA which are acid. DNA is acidic in nature, the histone proteins are the basic in nature. The histone proteins are the H1, H2A, H2B, H3 and H4. So among these, H1 is loosely associated with the DNA and the remaining involved in structural organization of the chromosome. So histone proteins are mainly associated in the structural organization of the chromosome, chromatin fiber, and it is arranged inside the chromosome. The non-histone proteins, these proteins include many enzymes such as DNA, RNA polymerases. This class of proteins are enzymes. That means here the non-histone proteins are include many important enzymes. The polymerase enzymes are seen in this. Next, eukaryotic chromosome structure. So, eukaryotic chromosome structure, how, that means how these parts DNA and proteins, how they are associated or arranged in the chromosome, means there are so many theories are proposed regarding this the chromosome structure. One of it is the folded and fiber model, which is proposed by Dr. Pran in 1965. According to him, this mode of chromosomes are made up of by the chromatin fiber about 230 angstrom units diameter. This chromatin fiber is produced coiling the single DNA, double helix and coils of DNA molecules stabilized by the proteins. The 20 angstrom DNA double helix is specially packed in protein to form fibrils. These fibrils further coils to form fiber of 100 angstrom units. This fiber is called type A. So, these fibers are closely packed inside the chromosome part, and here in this theory Pran was not explains the role of protein in the chromosomal organization. The next theory is the nucleosome model. So, nucleosome or solenoid model, this is proposed by, this model was proposed by Korenberg and Thomas. According to this, the chromatin is composed of a repeating unit called the nucleosome. The repeating unit called the repeating unit called the nucleosome. That is why it is named as the nucleosome model. Chromatid fibers appears to have a stinged by beads like structures. So, these nucleosome model, solenoid model, according to this model, the nucleosomes consist of a core particle of histones. These nucleosomes are linked together by a small fragments of DNA. This is called the linker DNA. With the help of linker DNA, all the nucleosome units are condensed and arranged and packed in a particular manner, that is the according to the solenoid model. So, this is the DNA packing. This picture shows the DNA packing in chromosome. So, nucleosome solenoid model, this model was proposed by Korenberg and Thomas. According to this model, the chromatin is composed of a repeating unit called the nucleosome. Each nucleosome have the histone proteins and the DNA material. These two are connected and form a unit called the nucleosome. So, chromatid fibers appear to have the sting of bead-like structures. So, because of these nucleosomes, the chromatid fibers appear to have a sting of bead structure. So, these nucleosomes consist of a core particle of histones. That means nearly 8 histone proteins are participated and they form a octamer like structure. This octamer is, these nucleosomes are linked together by a small fragments of the DNA. This is called the linker DNA. These nucleosomes are closely associated and form a solenoid like structure. So, these structures are arranged in the chromosome. So, in such a way the DNA material packed along with the linker DNA and arranged into the chromosome material. This is about the structure, types, chemical composition and structure of eukaryotic chromosome. Thank you.